So we've had some great talks on evolution so far. Nico and Sarab have really laid the foundations quite nicely. And I want to tell you, I think I must work more slowly than everyone else, because this is something I started like 7 years ago, thinking about this, most of this project, and it's almost published. So the first part of it, anyway, it's a collaboration with between my group and Marston Linehan, who's the chief of urologic oncology at NIH. And what makes one of the things Marston does that's really unique is he handles a very large fraction of the families who have von Hippel-Lindau syndrome, which is a hereditary syndrome with a deletion or loss of function in VHL that's inherited. And as a result, patients with von Hippel-Lindau syndrome get many kidney cancers, clear cell renal cell carcinomas during their lifetime. And what's been known previously is that these tumors are actually independent tumors. So the kidney that's been taken out there, you can see lots of tumors all over the place. Maybe 10 or 15 independent tumors can be seen there. And we know that, and there's been a bunch of papers showing that, because there'll be different VHL second hits in all these tumors, which is widely believed to be one of the earliest lesions that allows these tumors to progress. They'll have different chromosomal copy number behavior. There's been some exome sequencing of different ones, and they show no exome mutations in common. So there's been a lot of evidence that these were clonally independent in some way. And it's one of the few human cases in which you have a constrained genome in a constrained environment, yet multiple examples of tumors arising in that. And so you can we can start using tumors from these patients to think about how strong the constraints are on tumor evolution given a fixed genome and environment. Obviously, it's very difficult to separate those two because they're arising in the same person. But the effect is really very powerful, and I'll show you some numbers a little later on. What's important to understand here is that we have a pretty strong belief that the clear cell renal cell carcinomas, CCRCs, that arise in von Hippel-Lindau syndrome and those that arise in sporadic disease are quite similar, but their clinical presentation is quite different, right? So what typically happens is that because it's inherited, you actually know who's at high risk for having von Hippel-Lindau syndrome because their parents had it, or one of their parents would have it as well, right? And the fraction of de novo mutations is relatively low compared to the contribution from familial inheritance. Maybe 15 or 20% of new cases are sporadic versus inherited. But what you can see is that the sporadic cases on the left, they typically don't diagnose until much later in life. The people who have them are typically 50s, 60s, 70s, and the tumors are typically very, very large. The one that's pictured there is probably on the order of 10 centimeters or something, which is a massive tumor. You know, it's much bigger than a baseball. Whereas the ones that in hereditary clear cell renal cell carcinoma tend to be small because people are constantly being imaged, and the standard protocol is to image people to look for new tumors roughly every 6 months or so. And what they found is that as soon as they see a tumor, they don't go and operate right away. And the reason for that is because there's too many tumors. These patients would constantly be in recovery. They'd be having new surgeries every 6 months or so. And these are, relatively invasive surgeries that we're talking about. So the clinical treatment has actually been that you follow these patients, and if a tumor gets to 3 centimeters, then you operate. And when you go in and operate, then you take out all the tumors you can find. So at one time, you might go in and take out 3 or 4 or 5 or 10 or 15 independent lesions in one single surgery. And there'll be a range of different sizes from, as small as you can see, a ½ centimeter or something, all the way up to 3 or sometimes 4 centimeters. Sometimes, even on the regular surveillance, they'll have a tumor that goes very quickly from 1 centimeter to 5 centimeters. And so, a small fraction of tumors might be closer to the size you get with sporadic cases. But in general, these tumors are typically in the 1, 2, 3 centimeter range when they're extracted. And so the difference in size of the tumors is really quite marked here, right? We're talking a tumor that might be on the order of 1 billion cells versus a tumor that might be on the order of 1 trillion cells or something like that, right? I mean, that's a lot, obviously three orders of magnitude difference in what's going on. So Cancer Genome Atlas has published its initial study of clear cell renal cell carcinoma, and it recapitulated essentially what was known previously from earlier studies. that we have a very high frequency of mutation von Hippel-Lindau, VHL gene, the very first bar up there. Something on the order of 50% of cases have mutation of VHL. Another 10 or 15% of cases have methylation of one allele of VHL. And VHL is located on 3p21, and you can see more than 90% of the TCGA cases, the top bar in the lower panel there, have loss of chromosome 3p21. So what we have as a standard mechanism is loss of one allele of VHL, followed by loss of the chromosome on the other chromosome that carries a good copy of VHL. And the model typically is mutation followed by loss of heterozygosity. So you can see a number of other genes, PBRM1 and SETD2 and BAP1, which are all the same genomic location as VHL. They're all nearby. And they are expected to be tumor suppressors as well, and so the loss of 3p21 reveals these. So we have, on the order of 10 or so genes that are drivers from TCGA from a pretty good sized study, 385 cases, so that we've probably gotten most of the reasonably common drivers down to the few percent range. And you can see some very stereotypical copy number aberrations. Okay, so moving on to what we actually did now. Susie Fay in my group, who was a postdoc who's since moved on, and Marston's group identified a set of 6 individuals from which 42 tumors were selected. And the tumors are all, as far as we could tell, were all clonally independent. And we had a range of anywhere between 5 and 13 independent tumors per individual. Susie selected them to be sort of an equal mix of gender, smoking history, type of von Hippel-Lindau mutation. There's a relatively, sort of independent of the number of surgeries, a range of folks who were smokers and not, which is a known risk factor, a range of body mass indexes, and for all 6, we have blood as a normal control. For 3 additional tumors, we have normal adjacent kidney that we're pretty sure is pathologically okay. And you can see here, just in this number of surgery column, that there's a range of surgeries, right? So this 6-year-old woman, this is her 3rd surgery on her right kidney already. And she's had as I assume radical, it means her left kidney's already been taken out, right? So there's pretty substantial history for some of these individuals, but for this male, 28-year-old male here, this is their first surgery so far, and they've not had one on their left kidney yet. So Susie, we contracted with Illumina to do whole genome shotgun sequencing at roughly 40x, which is obviously a little lower than we'd like, but at the time financially expensive already. And Susie called mutations using Mutect in a relatively standard pipeline to identify. And so one of the things that was interesting to see is that there's quite a strong variation, just the absolute number of mutations per individual, right? So tumors from the same individual have a fairly good spread, but there's quite a strong bias that tumors from the same individual have a high concordance with the yellow individual, sort of the outlier there. If you look at that association with age, you can see that this seems to be mostly a function of age. The oldest 2 patients, the older the patients are, they have far more mutations. And what she observed here is that we have a couple outliers in these older individuals. This tumor turns out to be very low purity, probably like 20 or 30% tumor. And if you've only got 40x tumor sequencing, you're going to have a hard time calling mutations there. And so we expect that the number of mutations in this tumor is probably quite a lot higher. We just can't observe them. And this tumor actually turns out to be 2 overlapping tumors based on re-examination of the pathology. Once we say, well, why does it have so few mutations? So the correlation is quite strong. We were surprised that there wasn't a stronger correlation with smoking. We expected to see that smoking would contribute to the mutation burden here. It does not appear to. This is not adjusted by age, but I'm pretty sure that even adjusted by age, it'd be a very minimal correlation to smoking history here.
Sorry?
Yeah, of course.
These patients get metastasis?
So the clinical course is, if the tumor is smaller than 3 centimeters, they've never observed a metastasis. So in thousands of individuals, right? So once you get a 4-centimeter tumor there's some small risk of metastasis, like 5% or something. But if the tumors get to, say, 6 centimeters, then the risk of metastasis is very high. So the reason they picked this 3 centimeter threshold is basically they've never lost a patient because of that. Sometimes the tumors accelerate very rapidly and go from 1 centimeter to 5 centimeter over 6 months, and then they can't image constantly to look for these things. But no, because this is a relatively indolent disease, the sort of 6-month imaging is a strategy that works for the vast majority. So, I think most of these patients end up, not dying of kidney cancer. I think most of them end up dying from, eventually they have to have a kidney transplant, and so maybe they can't get an effective kidney transplant, which I guess you could call a consequence of the cancer, right? They no longer have a kidney. But it's not quite the same thing as the cancer killed you, right? It's just that they have loss of kidney function. So once they're on transplant, transplants are better than nothing, but not as good as having your own kidney. So just to confirm what we expected to be true, that these tumors are in fact independent. We looked at roughly the 40,000 mutations that we called across these 40 and more than this, like 100,000 mutations across these 40 tumors. And this is just a heat map showing them with the brighter the red, the higher the allele frequency. And what you can see is very strong stripes associated with individual tumors. And if you go in and look at, say, a region like this, where you get a whole band of sort of low minor allele fraction across a range of tumors, these almost always are sort of sequencing artifacts. And that, but it's a relatively rare phenomena in the data set. And, this is just blowing up a few of these things that look like they might be shared mutations across individuals. Here might be an example of 10 or so mutations that are somehow clonally related with the founding cells out of the more than a couple thousand mutations in that individual. Maybe there's 10 mutations or something that were foundational to the cells that derive both of them. And that's actually not too hard to imagine from just early development kind of point of view.
But doesn't that suggest then that they're not really independent? They do have a ancestral relationship.
So this is sort of what you mean by ancestral to me, right? So obviously all cells in the body came from an egg. So it's some very strong definition of ancestral. They have to be ancestral, right? And so I would argue that if these are real mutations, which I'm not even sure that they are, then we're talking about on the order of 8 mutations out of a couple thousand. Right, so it is a very early split, and we do see some evidence for things that look like mutations between tumor and blood going away when we use matched kidney normal, and so that could be something funny about the sequencing of the blood, that we missed a rare variant in the blood and we couldn't filter it out. Or it could be that they're the progenitors of the kidney, which I don't know exact number, but it's probably in the thousands or something of primordial cells, could contribute to that, right? And so you sort of expect there to be mutations even that early in development as you're putting up the foundation. So I think we're talking about extremely minimal early contribution, but it is a pretty robust way of assessing this behavior. So we obviously started this because we thought there might be commonalities between tumors in the same individual, right? If we hadn't found anything that was common between tumors in the same individual, we'd been very sad, because this would have broken our fundamental hypotheses. And so obviously the first thing to do is just start looking at mutation frequencies, and we can see quite strong biases, highly statistically significant biases in which mutations are showing up in which patients. And so the strongest one here is basically this individual, F28 green, so 28-year-old female who we've colored green for all concordance in the plots, has nearly twice as many T to G substitutions as anyone else in the population. These are relatively rare mutations overall, but she's got a lot of them, relatively speaking. You can see there's some wide variation in this blue patient here in the number of T to Cs. And C to Ts, which are relatively common, there's still statistically significant variation amongst those. So we're seeing biases in the mutational spectrum, but we don't know if this is environmental or genetic. It seems very plausible that it's either or both. And you know, I think that's something we're fundamentally interested in trying to examine. Don't necessarily have great ideas on how to do it, but open to suggestions. And then pushing this a little further, we've tried to look at the mutation spectra, sort of the things that Peter Campbell and Gatti Getz have published on, as well as many others, where we take the trinucleotide context. We have actually been very unsuccessful recapitulating the published signatures for clear cell kidney cancer in our data set. We can see trinucleotide contact biases in our data set, but they don't correspond to the published ones that are supposed to be present in kidney cancer. And so I'm not sure if we don't know how to do it, we're morons, or if there's fundamental differences in the way we've tackled this problem versus other groups. But you can see quite strong differences in, for example, the C to A, C to T spectrum here, which there's a set of tumors here that have higher propensity for. Here's T to A and C to T mutations that are higher in this group. And then here we have roughly C to G mutations, and then a very strong set of tumors that have T to C substitutions in a wide variety of contexts. And so there's clearly bias across the distribution, and there is bias across individuals. You can see all the patients who are yellow from one individual are on the left side of this dendrogram. All the green patients are on the right side of the dendrogram, and you know, with some straightforward I actually don't know the statistical method. I didn't look it up. I apologize. There's a p-value of roughly 10 to the minus fifth on this behavior, and by permutation testing as a separate way of checking this, we're seeing a very strong bias in the mutations, the spectra that we see across the individuals, and how they're organized. So it looks to us like, as you might expect, either genetics or environment is playing a pretty substantial role in the kinds of mutations that are showing up in tumors during evolution. But we were really surprised that we didn't see smoking, because some of these patients have a pretty substantial smoking history, and we don't see that, but it is a known risk factor for clear cell kidney cancer. Okay, so going back to the TCGA data, and I showed you this is a plot of the copy number where chromosomes are across the x-axis here, and patient samples are on the y. And using IGV, we can just ask, are individual chromosomes gained or lost? And based on whatever I told you, you would expect to see 3p loss. And so here's 3p. And you can see that nearly all these patients have loss of 3p. 91% of all the patient tumors have loss of 3p in this data set. Okay? You can see other stereotypical behavior gain of 5q. There's some reason to think that the 3p and 5q are fused together most often, so there's a mechanistic basis for this rearrangement. You can see loss of 14 here, right? So there's a stereotypical pattern, but this loss on 3q, on 3p is the standard thing that happens, and it's also what happens in our hereditary cancers, and this obviously was well known prior to this. But when we looked at this, we were immediately struck by one specific behavior, that this individual here, again, the sort of standout F28 green individual, in addition to the loss on 3p, which happens in all of her tumors, 10 out of her 13 tumors have lost 3q, okay? And only one other individual, or maybe two other tumors, this data is not very good, unfortunately, from copy number for this individual for some reason, have lost 3q. And if you go back to the TCGA data, you can see that loss of 3p is standard. Loss of 3q is actually relatively rare. Here we're sorting for it, and it's only about 10% of cases have lost 3q. So this is a relatively rare phenomena in the data set to lose 3q. And yet here we have an individual who's lost whole chromosome 3 10 out of 13 times. And so the p-value in that is astronomically small. There's a very strong bias as this individual is behaving differently in terms of chromosome loss. And our first thing, maybe she just throws chromosomes more easily, right? So, you know, whatever her genetics or environment are, is she's losing chromosomes more rapidly. But then you just look at the rest of the genome, and there's no increase in the rate of chromosome loss anywhere else. So it's not something that simple, right? And so we're sort of deeply trying to think about mechanisms based on that would be biased towards loss of a specific chromosome. We've got a bunch that we're trying to test, but haven't come to anything compelling yet. And so this bias here could be genetic, could be environmental. I think we favor genetics on this one for a variety of reasons. But remember, this is always the same chromosome that's being lost, because one has an inherited VHL allele, and so we're always losing the other chromosome here. Once you accept this result and you go in and look, there's actually some pretty believable other biases. Here there's 3 men. This patient has lost chromosome Y in 3 out of 6 tumors, which is a relatively high rate. Not enough given all the multiple testing you'd have to do all this that you'd believe it, but seems to be a bias there. The frequency of rearrangements between 3p and 5, or sorry, between 3q and 5q, as estimated by gain on 5q and loss of and retention of 3p. No, retention of 3q looks to be perhaps biased as well, with this individual showing it quite frequently. And so I think that there are going to be, with a bigger data set, you'd be able to see more biases in the chromosomal patterns that are happening here. And I think the vast majority of those will turn out to be genetic. And so, yeah.
Can you actually measure the fusion of 3 to 5?
Yeah, I think a lot of times it happens near the centromere, so I think a lot of times it's very difficult to pull out. Our group has historically been very bad at finding chromosomal fusions for whatever reason, so I think we'd be more than happy to share the data to someone who really thinks they're good at it, but we have not been very successful at it, and I think because it's typically near the centromere, we kind of, if we didn't see it, we couldn't really be sure that it wasn't just hard to see. So I don't know. Okay. So going back then to our initial question, the easiest explanation was for this behavior would be simply that the tumor that arose out that had lost whole chromosome 3 was a clone of some early thing, right? A very early event lost whole chromosome 3, and then all the tumors grows out of that. And we have, and so this is looking at all the mutations that are shared across the individual tumors here. And if that were true, you would expect that the 10 tumors that are in light green would share some set of mutations that are excluded in the dark green tumors on the outside here, right? That those would be foundational mutations that arose very early in the lineage, and because we have whole genome data here that does not show evidence of shared mutations, we really think these are independent losses rather than an early loss that set up a field defect or something like that. And the number of mutations here that you just would expect to see, 1 or 2 or 3 or 4 mutations, given how many we have. That are shared uniquely amongst here, and there's really no evidence for it at all. So I think the vast majority of these are probably sequencing errors or something like that. It's without checking them all, it's a bit of a hassle. But there's no reason to think that this is an early event. And so this is just commenting on the potentiality of shared stuff. Okay. So going back to what we know from TCGA, we've got this set of driver genes that are listed there. It's on the order of 10 or so. And since the clear cell paper came out, a bunch of groups have looked for other driver genes that are sort of pan driver genes. And so using the 120-ish so that are published in the PanCan paper, we wanted to ask, what's the evidence for driver genes in these tumors, and are we seeing the same driver gene happening in the same patient? Because that would be very interesting, right? If the constrained genetics and environment were pushing the same sort of driving lesions, that would be a very cool sort of constraint on tumor evolution. And obviously we were really hoping to see some effect like that. And instead what we saw was really odd to us. That when we looked at the tumors, we call the VHL a driver because we have loss of heterozygosity in effectively every one of these tumors, and we know they have one bad defect. So we know every one of these tumors is loss of function for VHL, and so every tumor here gets a VHL mark just for showing up, and the size of the tumor in 2 dimensions is drawn to scale here. So these are, this is like a 1.5 centimeter, 2 centimeter tumor. Here's a 1 centimeter tumor for each of the different tumors from all the different individuals. And Chris Ricketts with Marston's group then added some knowledge about how aggressive these tumors were based on pathological grading. Okay, so in the lighter colors we have grade 2, which was less bad than grade 3, and what quickly popped out at us is that the higher the grade tumors, they had a lot more driver mutations to them. And so this is looking at the clear cell SMGs listed 16 here. The PanCan SMG is 114, and all TCG, any gene that was ever called a driver in any tumor type, 250 or so. And so, this individual here, number one, he's developing a lot more aggressive tumors, relatively speaking. 4 out of 5 of his tumors are grade 3 versus, 13 out of 13 are grade 2 in this individual. So there is a patient-specific effect on grade, which is kind of interesting. But there's this corresponding effect on number of drivers here. Okay, and so these tumors have lots of driver mutations, seemingly, versus the vast majority of these things have VHL and nothing else. And so that was really puzzling to me because, I was taught, and people have shown the slide, that tumors arise because they have a hallmark set of deregulated pathways. And I've got tumors here, lots of them, with VHL and nothing else, and we can't find anything else. And that was really, really, I found it surprising. And it's not the first time we've seen that, but these are pretty simple tumors, relatively speaking. And so, because we only did 40x genomes here, there's a possibility we've missed mutations, right? Where maybe our mutation sensitivity is only 50% or something from 40x genomes. And so, maybe we really have drivers here that we just didn't sequence properly for whatever reason. So Susie went back and picked 10 tumors that sort of spread across grade, individual, and the number of mutations that were available. And we did exomes, reasonably deep exomes, 150, 200x exomes. So we were pretty sure that we'd pick up extra mutations. And when we did that, we actually found a lot more drivers. Okay, so the calls that were made in the genomes are on top, and the calls that were made in the exome are on the bottom. And so this big tumor here, I don't know, 6 centimeters or something, went from having nothing we could find other than VHL to adding a couple very plausible driver genes, Notch and NF1, and then maybe SPN or something, right? So the increased sequencing depth allowed us to see extra mutations, and that was true in the vast majority of cases here. But you can see that the grade 3s didn't typically add more mutations, right? So the grade 3s, when we sequenced them, they had roughly the same mutations as when we sequenced them later on. So this immediately suggested to us that this was not a sequencing depth problem, but it had something to do with the biology of what was going on. And so Susie looked at whether or not these mutations were clonal or not, and it was very obvious that there's a difference here. So on the left is just one tumor from the 28-year-old male. And this is varying allele frequency corrected by purity and copy number. And you can see that there are no predicted drivers in the clonal fraction here. But three of the drivers, that we see are showing up in the subclonal part. Okay, so it looks to us like the drivers here are all being contributed later in tumor evolution. Whereas you look at the grade 3, this is a relatively low, so it's a 65% tumor purity. The single driver here, which is in one of the TCGA SMGs for clear cell, is clonal. And so, just sort of looking through this pretty quickly, here's another independent tumor. Here's the clonal fraction. Here are the subclonal fractions. And again, the drivers that we expect to see are all subclonal in these individuals. Here's a grade 3. Here's the grade 3. Where it's a clonal driver. Here's another grade 3 where there are 3 plausible driver mutations that are clonal, a couple more that are right on the shift to becoming clonal, so they're at the inflection point. Here's a grade 2 where looks like there's 1 clonal driver and 1 subclonal driver. And so I think what this means is that these indolent low-grade tumors have acquired VHL and maybe not much else, right? Maybe all it takes to be a tumor is VHL and some epigenetic rearrangement, right? Not a lot of what we call classic driver events. That's one interpretation of the data, and I don't think it's the only one. So that immediately brought us back to the Gerlinger paper that had been that people have talked about already at the meeting. And I want to highlight one thing that no one's reminded everyone about is that one of the most compelling things about this paper is that different parts of the tumor have solved the evolutionary bottleneck with different mutations, right? And so we have 1, 2, 3 SETD2 mutations that are all, as far as we can tell, independent and in different cells, right? So that's they're at different sites, so they're not being constrained by the, but the tumor is solving this problem in 3 different ways in different clones, and that's quite striking. And so I think we need to pay attention to that. And so this is a Gerlinger paper on sporadic cases. This is a 10-centimeter tumor. This is a monster tumor that they've looked at, right? And so there's a lot of opportunities here for subclones to grow out. Swanton's group has also looked at a few hereditary tumors from von Hippel-Lindau cases, not quite as in depth. And they report actually something slightly different. They don't see the branched evolution that you show here. They did not provide a figure, so I had to make one, and I made it really lame. My apologies. But if you took this multi-region sequencing approach that they've drawn for this VHL patient, R4, R3, R7, R2, R6, you get a tree that looked like this, and they call this linear evolution rather than the branched evolution. And so there's, from review you can find examples like this where you've got serial replacement of a population as evolution goes forward versus branched evolution where you have different pockets of the tumor or intermixed populations of the tumor all coexisting at the same time. And I'll be honest, since I work on VHL syndrome, I'm not very happy about this result because I want VHL syndrome to be as much like hereditary or sporadic cases as possible. So I found this a little problematic, but I think our observation about grade sort of implies that maybe this is a little more subtle, and so we're in the process of trying to reanalyze the Swanton data in a slightly different context, and I hope that we'll be able to provide some deeper insight. So I just want to take some of the things we've seen and extrapolate them a little bit about what they mean for evolution, because I think sometimes these principles get lost. And when we see branched subclones, there's a lot of possible reasons for that. And when we see linear evolution, there's a lot of possible reasons for that. And so for example, if you have multiple subclones in a branched tree, one hypothesis is that they could be spatially segregated. Over here is one branch, over here is another branch, over here is another branch. If they're not spatially segregated, it has different evolutionary implications, but it doesn't mean that they're not important things to take home. And so one possibility is that these things are happening late, and the tumor is evolving, and then they're acquiring mutations, and they grow out. Another possibility is that mutations in different compartments of the same tumor are actually synergistic or codependent, right? And so what's really important, and Saurabh touched on this, is that the fitness landscape is not constant, right? That you can't just say that this initial tumor has these mutations and its fitness is X and it's going to grow on and forever grow at some fixed rate. That's almost certainly untrue, right? The environment changes as a function of time. And because of that, everyone needs to remember that the fitness of an organism, or in this case a cancer cell, is dependent on its environment. With a change in environment, the fitness can radically change. And so the behavior of subclones is highly dependent upon what you think the fitness environment looks like. And I think this is a ripe area for study. Don't have any great ways of tackling it, but for the most part, we don't have ways of measuring changes in fitness over tumor evolution, other than the case where you apply chemotherapy and everything changes, right? Where you can know what the fitness change was, you can take away the fitness change and see if things revert back. And so I think there's some things to pay attention to here. And so this linear model, which we've seen in things like AML, where you get serial replacement of the population, is usually under a constraint where you've got an exogenous selective force that you've applied, right? And it's less clear whether or not the linear model that Swanton's reporting in VHL syndrome actually makes sense from a biological possibility. It's possible. It is possible that the tumors grow a little faster, and differences in the initial fitness population could favor certain differential behaviors. And I just want to. No one's showing something like this, so I think it could be instructive that if you've got a centimeter tumor and you have the sort of a typical model would be 3 new mutations per cell division, that has 1.5 billion mutations in it, right? So an enormous possibility of all the mutations that are allowed in the human genome have already happened in that tumor. And as you get bigger and bigger, the number of cells is rising with the cube. You get huge numbers of potential mutations showing up. So, you know, if you think about number of ways to make a null mutation in set D2, when is the likelihood that there are multiple set D2 mutations in a population of 1 million cells, 10 million cells, or 100 million cells? And actually, I think somewhere around 10 to 100 million cells, there's an inflection point where you actually expect multiple set D2 mutations. And if they have even a transitory, rapidly different fitness behavior, they could rapidly take over to at least co-compete in the population. Their fitness might slow down later on, but I think this is something that we need to pay attention to. And you know, I could draw much smaller tumors than this, but we have enormous tumors with enormous genetic complexity going on, and so I don't find it too surprising that we have genetic selection for subclones because of the diversity. So I more or less covered this, but I did want to sort of question whether or not there's even a difference between linear and branched evolution. Linear and branched evolution, and you know, they're sort of posited as being different things, but I'm not sure they really are. I think they're different. I think they're different views of different constraints on the same system, right? It's all evolution. So I'm not sure what to take over it. But like I said, we are looking in the VHL data, exome data from Swanton to see if we see, for example, subclones. And actually the evidence so far is we are seeing a lot more subclones than were reported in that paper. Okay, so in my 5 minutes, I wanted to get to sort of the last question of, why do these tumors have such few drivers? Right? I mean, I told you all about these hereditary clear cell renal cell carcinomas that have, on average, 1 driving mutation that we are smart enough to identify. And if you look in the TCGA data, here's the TCGA data. 20% of the tumors there, which are mostly grade 3 tumors, have no drivers that we're smart enough to identify, right? Things that look like we understand what's going on. And it gets worse for some tumors. So here's data from papillary kidney that we published that just came out this month, I guess, or last month. And this is looking at type 1 and type 2. This is the copy number profile. There's a very large. This is patients on the y-axis here, genome on the x. You can see that a lot of these patients here have a very stereotypical chromosome, single copy chromosome gain pattern. As a geneticist, I want, you know, a point mutation in RAS or, you know, some really obvious strong driver. Gain of chromosome 3 is a very uncompelling, or gain of chromosome 7, sorry, is a pretty uncompelling driver to me. I'm like, really? That's the best you can tell me about what's making this a tumor is it has gain of chromosome 7. And in papillary, with a reasonable number of cases, 200 or 180 or something, there's a relatively small number of drivers. Although it's about the same as 157. But their frequency of mutation is very low. Okay, and if you then go and look for cases that have no drivers in papillary, it's huge. And almost half of the type 1 tumors that we sequenced have nothing other than chromosome 7 gain that we can really believe as a driver. And you know, either we're going to have to be really good about understanding how chromosome 7 gain drives papillary kidney cancer, or we're missing a lot here. And so I think we have a big challenge in a bunch of tumors, particularly the relatively indolent early tumors, which both clear cell renal cell carcinoma and papillary start off as. And so, you know, these are very good outcome tumors, and we call them cancers and they look like cancers to pathologists, but they don't have drivers, which is, I find, unsatisfying. So I think it's a big challenge to the computational group is to figure out what the hell is going on in here. All right, and so on time, more or less, I will stop there. And so Susie, Faye, and my group in Asia, Mitchell, did the vast majority of work. Katie is the one who actually got us the samples, which took, you know, that when I said it took us 7 years, it took us like 4 years to get samples, working with the federal government. And then Marston and Kathy and Chris at NCI. So, and then the whole TCGA teams. Thank you. Roland.
I'm interested in this branch versus linear evolution, right? So, are the sporadic tumors more aggressive than the hereditary, or is there any difference?
They tend to be. But they also tend to be found much later, right? So the hereditary tumors we find because we're looking for them, and the sporadic ones we find because you have a clinical problem. You can't blood in your urine or something, right?
I'm just curious because we saw in ovarian cancer on a small sample size that a very strongly branched evolutionary trajectory typically is associated with poor survival and lower survival.
So this is high-grade serous ovarian cancer. Yeah, I mean, so the outcomes in ovarian are so awful. I'm not sure what to do about that, right? But I believe it. I really do believe it. I think that, and there's a fair bit of evidence that more genetic complexity is a bad prognostic feature. But particularly the high-grade serous ovarian cancers have such a nasty outcome that I'm not sure what to do about it exactly. But yeah.
No, but I was just wondering if you have any data for between the linear and branched for
like I said, I don't think it's a real distinction. I mean, I think
it's both evolution, right? But like you said, it's kind of the same underlying process, but then different selection pressures maybe, or different more clonal expansions on one side, or there are different aspects of the same thing, of course, but that doesn't mean they're not different in a qualitative sense.
Yeah, I think it has to do with selective pressure, right? That you'll get a branched architecture under certain selective kinds of pressures and under certain population sizes. And you'll get a linear model when the population size is really small, for example, right? Or when there is, so there's been a fair amount of literature on the idea of clonal expansion without death, right? So if you have death associated with a behavior, and you're removing cells from a population, you could get things that look much more linear than if things don't actually go away, they just peter out, right? Those will look very different, and so I think it has to do with the kind of selective pressures that are going on. But my bias is for clear cell, it's all going to be branched when we get down into it. But I think in other cases where you have a very strong selective pressure that can remove things from the population, then you get this linear behavior.
Just a quick follow-up. I mean, for your estimates of how many mutations are in these tumors of 6 centimeters.
Yeah, wildly speculative.
But there you're assuming that the cells you're seeing are the ones that were produced during the growth of the tumor, right? And if there was death?
Sure, it could be even higher. Yeah.
It would be an even greater number.
Absolutely true, yeah.
Do you know whether death occurs during the growth of those tumors? Or, I mean, it seems to be like a really important question for evolution.
It is almost certainly tumor dependent. There's, I think, a reasonable evidence that some tumors, for example, have higher rates of chromosome missegregation, right? Their genomes are more aberrant. As a result, they're more likely to throw off chromosomal genotypes in their daughters that are not what the parental genotypes were. And that the fitness of those aberrant genomes is much, much lower. So whether or not they die versus just stop growing, I don't know how to answer, right? But it certainly wouldn't be the case that you would expect that the effective fitness of a cell of all of its daughters is equal to the parent, right? Which is a slightly different way of looking at death, and I'm not sure how that would play out exactly. But if two daughters of a cell, one has a fitness, the parent has a fitness of one daughter has a fitness of 1, the other daughter could have a fitness of 0, right? And even though it never divides again, it never propagates again, and so it doesn't contribute. So in that case, the number of cells would effectively be wouldn't actually matter that had happened. So it would only matter if there was death. And I think in some tumors there will be, and others there won't be. But I don't have good numbers. Yeah.
In your green patient with chromosome 6, entire chromosome 3.
3, yeah.
So I didn't quite follow the argument for why you think there's an independent.
So if they were dependent, right, if they happened early in tumor evolution, then we would expect to see some set of mutations that happened in the lineage of those early cells. And unless it was the very first cell of the kidney or something like that, before any new mutations had shown up, we would expect to see a shared set of mutations in those tumors that all had chromosome 3 loss and not in the ones that didn't have chromosome 3 loss, right? We'd expect some number of mutations. We can't prove that it's not the earliest thing we can measure, but because we don't see a set of mutations that are explicitly shared, we take that as you know, from our work on timing, we would say this is the earliest 0.1% of all events that could have happened, right? So ruling it out explicitly is impossible, but making a statistical argument about when it would have had to occur, I think, is pretty compelling.
Okay, let's thank Paul again.
Thank you.